Okay, today we're gonna move on to the next one, which is Endospore stem, lab 4. But let's just review what we have. Lab 3, the gram stem results. So what you have? We got E. coli. So what do you think of the E. coli's gram stem results? Ideally, it's gram negative, so pink color. It's lods, and we can say it's a single, relatively short lods. That's E. coli. What is E. facilien? E. facilien, Enterococcus facilien. That should be ideally gram positive. It's a coccile. It's a chain arrangement because originally it's belonging to Streptococcus. It is a pathogen surrogate. Okay, that's what we very briefly review lab 3. Okay, lab 4 today. We're going to do Endospore staining. So first thing first, there are two major bacteria culture or strains or genomes has Endospore, which is Bacillus and Clostridium. And the Bacillus could be Bacillus subtilis or Bacillus cereus. Clostridium, it could be Clostridium perfringens, could be Clostridium tetanus, could be Clostridium difficile, it also could be Clostridium botulism or botulinum. Clostridium is biosafety level 3 pathogen. We cannot do here. It's an endospore forming anaerobic pathogen, strict endospore forming. It will cause big problem. We'll talk about it in the lecture. We cannot do here. I believe in the United States, in the food science area, the lab in University of Minnesota Twin City can do. I do not think even in Virginia Tech or some of the nearby land-grant institutions they can do biosafety level 3 Clostridium. So we cannot do here. We will do is Bacillus. So today what you're going to have is Bacillus subtilis on the slant. Okay, it's the same thing, still a smear, but it's easy to grow on the slant, so that's why they do on the slant. So that's a very basic information. The species is Bacillus subtilis. Okay, second thing, we want to talk about the endospore. So what is endospore? Endospore usually it is for resistant to stress environment, like dry, like desiccation, like UV, like radiation, all kinds of unfriendly environment for bacterial growth. If bacteria can form in those spores, they can protect them. So it is a protection structure, not has reproductive function, which means the mother cell has endospore does not necessarily mean the daughter cell will automatically has endospore, because the endospore is major for protection. Now, what does the structure looks like? It is a multi-layer structure. Maybe one more. We'll see. Okay, the outside we call exorium. Then with this inside area right here, we call it a coat. Then we get here is called a cortex. Cortex very similar to the cell wall by polysaccharides. Then you go here, you have cell wall. Then you go here, you have cell plasma, or we say membrane system. Now, these only the structure. When you see the structure, you can see it's multi-layer, rigid structure. But what's more important for protection is right on the center. There are two important chemicals. Number one, calcium DPA. Diperconic acid. Second, very small acid soluble DNA protein. Because of these two structures right in the middle, make it resistant and protected for the harsh and stress environments. Okay, this is the key thing. So that's a basic thing for the Endospore. Now, why we have to do Endospore? We mentioned about the function to do the Endospore is there Endospore? So presence, absence. Second, where is the Endospore? Which means the location. And we talk about in the lecture already, the location could be center, it also could be end, we call it a terminal, like you go to the airport. It also could be right here, sub-terminal. And the location, it is also genetic dependent. So today you see the bacillus, it may be subterminal. Some Clostridium, it may be terminal. Other Clostridium species may be center. Okay, so that's why we need to do the endospore staining. We'll find is there endospore and where is the endospore. And the location is genetic dependent. Okay, how to do endospore stain? So the staining technology is special. But majority of that are similar. You get a glass slice. Today only going to do one. Okay? Right in the middle you draw a big circle. On the side you write BS. Bacillus subtilis. That represents Bacillus subtilis. Also could be Bacillus cereus. Okay? Then you flip it over. You do smear preparation. I'm going to not go detail of this one because you already know how to do. Is that right? You add 1 drop of the water, flame the loop, then you add bacteria, make a thin, averagely covered layer. It's called a smear preparation. Don't forget, air dry, 3 to 5 minutes, heat fixing. Okay, once this is done, we are moving to a major step right here, is the staining. What's a stain we'll have to use? Very special dye called malachite green. This is also antibiotics. It could kill lots of the bacteria. So once you have a glass slides ready, we will do two different methods. It's malachite green, just middle couple of drops. And remember, we said the structure is rigid. So the basic dye is difficult to penetrate. What we have to do? We have to use a special method called heat steaming. This is not heat fixing for 5 minutes. And there are two methods to do. One way is half of you will work with rayon. You're going to put into a water bath. The water bath is around like 80 degrees Celsius. So the water, the heating comes from the bottom to heat it. That's a heat steam. Then another half of you gonna work with me. This is the little difference is we will flooding with malachite green. What means flooding? The whole surface you're gonna cover malachite green, and then we're gonna use Bunsen burner to do the flame on the surface back and forth for 5 minutes. At this moment, once you finish, it's very hot. So you're gonna cool down, you rinse, and don't forget the counter stain with saffron. So, what the Endospore gonna looks like ideally? You will see majority of these are pink, the background. They are a big chunk of blue, a green there. That's Endospore. Or some of you see a big circle with white color in the middle. Those are Endospore. Could be two different results. I don't know. We'll see. Or could be have both. So that's Endospore. It should be very beautiful. We did the last couple of years. Okay. Last thing. Endospore stain and the gram stain. There is one difference. Gram stain, we have to use a very young culture, because we want the cell wall structure is excellent. If the culture is older, the bacteria start to die, start to cracking down, what will happen? The peptidoglycan already loosened, so you're not going to hold on crystal violet. It's all going to stay gram negative, we call it a gram variable stain. In the opposite, Endospore, we want them to be forming in the harsh environments. So basically the bacteria also needs to be older. So Endospore use older culture. Basically more than 48 to 72 hour culture. Could be even a week. You get lucky, the Bacillus subtilis what we have is growing really fast, and it will have Endospore easily. So that's a species we want to use. It's only talk about 36 to 48 hours we're having those spores. But generally speaking, most of the species need took 3 days to grow in the lab and then to form in the endospore. Okay, so that's what we have. Okay, today is a little bit different. I'm not going to show you the steps from here because you already did. You guys, everybody, work independently. Have a glass slide to prepare smear, add water, add Bacillus subtilis, which is on the tube here, on cue heat fixing. Then you have your hands on, said I'm ready. We're gonna put on the rack in these two bench. So these two benches you work with me, we use a Bunsen burner to do. And these two benches you work with TA and Rion at the what's the name? Payton and the Rion at the back. You guys using water bus. The difference is water bus and malachite green is only a couple of drops. As long as you cover the circle. Here we need to use lots of malachite green. Okay? So hope we have enough. So then you go ahead to do it. And if you get ready, I'll show you how to do the heat steaming.
Okay, one more thing. In the slides, make sure you write your bench